Methylation, the role of nutrient deficiency in the human body. Methylation is a crucial biochemical process that occurs in every cell of the human body. It involves the transfer of a methyl group, one carbon atom and 3 hydrogen atoms, to various molecules, such as DNA, proteins, and neurotransmitters. This process impacts a wide range of biological functions, from DNA repair and detoxification to hormone regulation and brain function. However, when nutrient deficiencies disrupt methylation, it can have profound effects on health, leading to various chronic conditions and increasing the risk for diseases. In this video, we will explore how methylation works, the essential nutrients involved, and what happens when these nutrients are deficient in the human body. How methylation works. Methylation is often referred to as a molecular switch because it regulates the activity of many important molecules. One of its key roles is to maintain proper gene expression. DNA methylation, for example, adds methyl groups to DNA, influencing which genes are turned on or off. This helps control cellular function, development, and even aging. Beyond gene expression, methylation is also vital for detoxification. The liver relies on methylation to neutralize harmful substances, including toxins and heavy metals. Brain function, neurotransmitter production and balance, critical for mental health and cognitive function, depend on proper methylation. Cardiovascular health. Methylation helps in breaking down homocysteine, a compound linked to heart disease when present in high amounts. Immune function. It helps regulate the immune system's response to infections, autoimmunity, and inflammation. For methylation to occur efficiently, specific nutrients act as cofactors or building blocks in the process. Essential nutrients for methylation. Certain vitamins and minerals are essential for proper methylation, including 1. Folate, vitamin B9. Folate is required to generate methyl groups for DNA and RNA synthesis. It is especially crucial for cell division and development, particularly during pregnancy. 2. Vitamin B12. This vitamin works closely with folate in the conversion of homocysteine into methionine, which is necessary for DNA methylation and protein synthesis. 3. Vitamin B6. Also involved in homocysteine metabolism, B6 helps convert it into cysteine, a precursor for glutathione, a major antioxidant in the body. 4. Choline. Choline is another methyl donor that helps with the synthesis of acetylcholine, a neurotransmitter involved in memory and muscle function. 5. Magnesium. Although not a direct methyl donor, magnesium acts as a cofactor in the activation of enzymes responsible for methylation processes. Nutrient deficiency and its impact on methylation. When the body lacks these essential nutrients, methylation can be impaired, leading to several potential health issues. 1. Cardiovascular disease. A folate deficiency, B12, or B6 can cause elevated levels of homocysteine, which damages the inner lining of blood vessels and promotes plaque formation, significantly increasing the risk of cardiovascular disease and stroke. 2. Cognitive decline and mental health issues. Methylation plays a vital role in neurotransmitter regulation. Insufficient levels of B vitamins, especially B12 and folate, can lead to mental health disorders like depression, anxiety, and cognitive decline. Impaired methylation has been linked to neurodegenerative diseases, such as Alzheimer's and Parkinson's. 3. Pregnancy complications. Folate deficiency is one of the most well-known causes of neural tube defects in developing fetuses. Adequate methylation during pregnancy is essential for proper fetal development, preventing birth defects and ensuring normal cell division and tissue formation. 4. Immune dysfunction and autoimmunity. When methylation is impaired due to nutrient deficiencies, the immune system may become dysregulated, leading to increased inflammation or an inappropriate autoimmune response. This imbalance can contribute to conditions like rheumatoid arthritis, lupus, and multiple sclerosis. 5. Cancer risk. DNA methylation regulates gene expression, particularly the activation of tumor suppressor genes. When methylation is disrupted, due to a lack of folate or B12, these tumor-suppressing genes can become inactivated, leading to an increased risk of cancer development. Recognizing and addressing nutrient deficiencies. Symptoms of nutrient deficiencies related to methylation can be subtle or severe, depending on the individual and the level of deficiency. Common signs include fatigue, cognitive difficulties, poor immune response, mood swings, and cardiovascular issues. To support proper methylation, it's essential to ensure that your diet includes foods rich in key nutrients. Folate, leafy greens, spinach, kale, legumes, avocados, and liver. Vitamin B12, animal products like meat, fish, eggs, and dairy. Vegans or vegetarians may require supplements. Vitamin B6, poultry, fish, bananas, and chickpeas. Choline, eggs, fish, chicken, and soybeans. Magnesium, nuts, seeds, whole grains, and leafy greens. In some cases, a doctor may recommend supplements to address deficiencies, particularly for individuals with absorption issues or increased needs, such as pregnant women or older adults. If you found value in this video, please give it a thumbs up, like and subscribe to support this channel. Please share across social media platforms to reach a wider audience. Maintaining methylation for optimal health. Methylation is a fundamental process in the human body, impacting everything from heart health to brain function. A diet lacking key nutrients like folate, B12, and B6 can disrupt this critical process, leading to a range of health problems. By ensuring an adequate intake of these nutrients, we can support healthy methylation and protect against potential deficiencies and related diseases. As always, it's important to consult with a healthcare provider to assess individual nutrient levels and determine if supplementation is necessary. With the right balance of nutrients, you can optimize methylation and promote overall well-being. How can one test for nutrient deficiencies related to methylation? Testing for nutrient deficiencies that impact methylation typically involves a combination of blood tests and genetic screenings. Some of the most common methods include: 1. Homocysteine test. High levels of homocysteine in the blood can indicate poor methylation due to deficiencies in vitamins B6, B12, or folate. This is often the first test used to assess methylation issues. 2. Vitamin B12 and folate tests. Blood tests can measure the levels of B12 and folate directly, helping to identify if these essential nutrients are deficient and impacting methylation. 3. Methylmalonic acid test, MMA. Elevated MMA levels can suggest a deficiency in vitamin B12, as this compound builds up when B12 is insufficient. 4. Genetic testing, MTHFR gene. Genetic variations in the MTHFR gene can impair the body's ability to methylate properly. Individuals with certain MTHFR mutations may have difficulty processing folate, leading to methylation issues. A genetic test can help identify these variations and guide treatment. 5. Comprehensive nutrient panel. This test assesses a broad range of vitamins and minerals, including B6, B12, folate, magnesium, and other nutrients that influence methylation. It provides a more comprehensive view of your nutritional status. By working with a healthcare provider, you can undergo the appropriate tests to pinpoint any deficiencies related to methylation. What are the best dietary sources for choline and magnesium, and how can one ensure adequate intake? Choline and magnesium are essential for methylation and overall health, and ensuring adequate intake of these nutrients can be achieved through dietary sources. Best dietary sources of choline. 1. Eggs, especially the yolk. A single egg yolk provides a significant portion of the daily choline requirement. 2. Meat and poultry. Beef liver, chicken liver, and lean meats like chicken and turkey are excellent sources. 3. Fish. Cod, salmon, and tuna are good sources of choline. 4. Soy products. Soybeans, tofu, and soy milk offer plant-based choline. 5. Cruciferous vegetables. Broccoli, cauliflower, and Brussels sprouts contain moderate amounts of choline. To ensure adequate choline intake, include eggs, meats, or fish regularly in your diet. For plant-based diets, incorporate soy products and cruciferous vegetables. Best dietary sources of magnesium. 1. Leafy greens. Spinach, kale, and Swiss chard are high in magnesium. 2. Nuts and seeds. Almonds, cashews, pumpkin seeds, and sunflower seeds are excellent sources. 3. Whole grains. Quinoa, brown rice, and oats provide magnesium along with fiber. 4. Legumes. Lentils, chickpeas, and black beans contain good amounts of magnesium. 5. Fish. Salmon and mackerel offer both magnesium and healthy omega-3 fats. 6. Dark chocolate. Contains magnesium and can be an occasional indulgent source. To ensure adequate magnesium intake, aim to include a variety of these foods in your daily diet. A good strategy is to consume a mixture of leafy greens, whole grains, and nuts regularly. Are there specific supplements recommended for individuals with impaired methylation due to nutrient deficiencies? Yes, there are specific supplements that are often recommended for individuals with impaired methylation. These supplements help ensure proper levels of nutrients crucial for methylation. 1. Methylated folate, 5-MTHF. If you have difficulty converting folate into its active form due to an MTHFR mutation, taking methylated folate, 5-methyltetrahydrofolate, is recommended instead of synthetic folic acid. 2. Methylcobalamin, active B12. Methylcobalamin is the active, bioavailable form of vitamin B12 and is often recommended for people with impaired methylation or absorption issues, for example, in people with pernicious anemia or MTHFR mutations. 3. Pyridoxal 5-phosphate, P5P. This is the active form of vitamin B6. It supports homocysteine metabolism, helping to prevent its buildup and supporting methylation processes. 4. Choline supplements. Choline bitartrate or alpha-GPC supplements can be used if dietary intake is insufficient. This is particularly helpful for individuals with higher choline needs, such as pregnant women. 5. Magnesium supplements. Magnesium glycinate or magnesium citrate are commonly recommended forms due to their high absorption rates and gentle effect on the digestive system. These are ideal for those who struggle to get enough magnesium from diet alone. 6. SAMe, S-adenosylmethionine. SAMe is a compound involved in the methylation process. Supplementing with SAMe can support individuals with methylation deficiencies, especially in managing mood and brain function. 7. Betaine, trimethylglycine. Betaine is another methyl donor that helps reduce homocysteine levels and supports liver function. It is often recommended alongside B12 and folate supplements. Before starting any supplements, it's essential to consult a healthcare provider to determine the right type and dosage based on your specific deficiencies and needs. Genetic factors, diet, and individual health conditions all play a role in determining the most appropriate supplements. If you enjoy this video, please give it a thumbs up, like and subscribe to our channel for more self-improvement videos. Comment below to tell us how useful you found this video. Remember to share on various social media platforms to reach a wider audience. Click the notification bell to get notified anytime we post a new video. Thanks for watching, see you in the next video.